Hello, this is Ms. Submingo with another semi-long video to help you guys for your AP exam. Again, I'm doing this for you, so don't get mad at me. This particular video, we're going to do a little bit more about genetics. The main points we're talking about is how do they know DNA was the one that does all the genetic material. We're also going to figure out how DNA copies itself, and what does DNA, how do you get DNA to actually do something. So those are the main things we're going to cover today. So just a little background, there are some experiments that you need to know. Griffith, one of the most famous experiments to figure out how things were inherited. So if you could see or zoom in or look at this, here's a mouse, and we have these, they call them S cells. Let's just say S representing a variable. And we give these S cells, we inject the mouse with these S cells, and the mouse dies. Then I take R cells, which I know are known to be good, I inject the mouse, and nothing happens, the mouse is fine. The third trial, I take these S cells, which originally killed the mouse, and I heat kill them. So basically they have this bacteria, so I heat it up so like the bacteria is dead, inject it into the mouse is alive. But the interesting part is this last trial where they mixed in my living R cells and the heat killed S cells. So when I injected this, the mouse lived. When I injected this, the mouse lived. But when I injected a combination of these two, the mouse died. And you're like, why does the mouse die? Like when I injected them both, they were healthy. But apparently, you know, in the living cell, the dead cells gave some of its genetic material to the living cells and was able to infect the mouse, and it died. And eventually it had living S cells in it. So the conclusion was that the living R bacteria transformed into the deadly S bacteria by some heritable substance. At this point, they didn't know what DNA was. The next famous experiment is by Oswald Avery, and he discovered that that thing that is transformed, or the thing that is being passed down, the thing that was transforming these S cells was DNA. Like DNA was the molecule that is passed down, and is passed down from parent to offspring. And it's before they didn't know if things were passed down or not, and now they knew something was passed down, and they knew that that thing that was being passed down was DNA. The other most famous experiments by Hersey and Chase. They have these bacteriophages. They look like spiders almost, and they're a virus that infects bacteria. And they did this experiment, and I don't want to explain the whole thing, because again, I want to make this as quick as possible, but they found out that it wasn't just DNA that was part of this heritable thing, but it was DNA and protein. So they did this experiment, and just know Hershey and Chase, they found out that it was DNA and protein that is passed down. So the conclusion is that DNA entered infected bacteria, and DNA must be the genetic material. So DNA and protein together, they make chromosomes. So that's part of the thing that they found out. Chargoff, another experiment that you need to know. Chargoff rules, you already know, again, reviewing A, T, and G and C. You know that A pairs with T and G pairs with C. Before they knew that, they just knew that every time that they had A's, they had the same amount of T's. And they knew that every time they did this and they found a G, they had the same amount of C's. They didn't know that they pair, they didn't know how DNA looks like. All they knew is that the ratio of A and T's were the same, and G and C is the same. So when you hear Chargoff or Chargoff's rules, it basically says that the same amount of A's is the same amount of T's, same amount of G's is the same amount of C's. The structure in DNA was found out by, first, Rosalind Franklin, so a female scientist. She found out that it was kind of in a shape of an X, which is representative of that chromosome that we see. So she was the first person to take a picture of DNA. Watson and Crick basically took the information from Chargoff's rules, the picture from Rosalind Franklin, and figured out that it was a double helix, like a twisted ladder, where the backbone of DNA is sugar and phosphate, and the rungs, the inside of DNA, if it's a twisted ladder, is the nitrogenous base, like A, T, G, or C. So DNA, again, on the outside, as you can see here, this is the nitrogenous base. There's A and T, and there's G and C. And here on the outside, you see where it says sugar, and sugar here. This is that phosphate, sugar-phosphate backbone. A and G are considered purines, and T and C are called pyrimidines. And again, a purine is paired with a pyrimidine. So A and T, so purine and pyrimidine, they connect together. So A and T goes with each other, and G and C goes with the other. A and T is a double bond, and you can see here G and C has like a triple. You can also see it here. Here's two bonds, here, and here on this picture for C and G, you see triple bonding. The structure of DNA, just to continue, when they're bonded, the A, T, and G and C, those bonds are hydrogen bonds. So I said G and C have a triple bond, that's hydrogen. A and T have a double hydrogen bond. Here's a picture of a diagram. Here is what it looks like chemically, and this is what it looks like if I did a 3D model of DNA. They are anti-parallel. So remember I told you about 5 to 3. This one is called the 5N, and this one's called the 3N. You may be wondering, like, how do you know this is 5 and what's 3? What does that mean? Basically, there's 6 carbons on the sugar, and the 5th carbon is at the end of this one, and at this end, the 3 carbon is at the end of the sugar. So basically, scientists didn't know how to label the sides of the DNA, so they found out if this sugar has a 5 here, we'll call that the 5 end, and if this sugar has a 3 at the end, we'll call this the 3 end. So when they say they're anti-parallel, one DNA goes from 5 to 3, and the other strand, the other backbone, goes from 3 to 5. Anti-parallel. Here's a zoomed-in version of what it looks like for chromosomes look like. So when we talk about the X of a chromosome, it's this thing right here. It's basically DNA, this long ribbon, and it's twisted around these proteins called histones. And they wrap around the proteins like so. It looks kind of ugly, but that structure is called a nucleosome. And a whole bunch of these nucleosomes make that chromosome, the chromosome structure that you're familiar with. And here's the second part of it. So again, here's the beginning. I wrap and wrap and wrap and wrap and wrap, and it looks like this X chromosome. That's how DNA and chromosomes, how they're related. So just to compare different DNAs, we've done this a lot, but you should know eukaryotic DNA is double-stranded and it's linear, whereas prokaryotes, they have a circular DNA with one chromosome, and usually we have more than one chromosome. We have, again, we talked about this before, we have 23 pairs of chromosomes, but every animal or organism that's eukaryotic, they have a different number. Ours is in the nucleus, theirs in the cytoplasm. They have no histones, which is those proteins that it's wrapped around. We do. And then they get supercoiled DNA when it's time to replicate. Ours is chromatin, if you remember. Replication is what they call semi-conservative. So if you see here, this is a molecule, and when they separate the strands, they